Hello, everyone. Welcome back to Heng Education, where we share the knowledge of biology. So today, in this video, I will be sharing how to answer Cambridge IGCSE Biology Paper 6, which is also known as the alternative to practical. So which is the, let's say, practical paper. And I will be sharing how to answer for February, March 2019. And you can also check out my other video where I share how to solve the multiple choice question paper, which is paper 2-2, February, March 2019 as well. So let's get started. I have already go through this paper and I have answer already written. So I will be going over with you all together how to approach the answers and how to get the correct answer and also how to draw graph and the calculations and everything. So the first one is that the student investigated the effect of two different types of washing powders on stained clothes. So from this statement already, we can observe that the effect of two different types. So it's mean different types of washing powder. So it's already mean independent variable, and it will be study on the stained cloth, right? And then there will be like information provided. So one was biological and another was just detergents, right? So it's mean that they are like two types. So one is biological and another one is non-biological. So it's already saying us that those are the independent variable. And also the student will provide it with 4 pieces of dry clothes. So this can be the constant that has been stained with the same food. This is very obvious. They mentioned same food. So it will be the constant variable. And they give a solution of biological washing powder, which is as labeled as bio, and non-biological washing powder as non-bio, and also distilled water as bio. And then a marker pen was used to divide a white tile into 4 sections. So 4 sections, each one will have bio, non-bio, water, and not washed. So a piece of stained dry cloth was placed on the non-washed section of the white tile, and the other were placed into each solution labeled bio, non-bio, and water. And in step 4, I would suggest students to pay attention to the every step because they will come back and ask you like the error or improvement or why this result is not valid or why this is not very accurate, something like that. So in step 4, it's mentioned that one glass rod was used to push the pieces of cloth into liquid in each beaker. So it's saying that only one glass rod was used to dip the cloth into different solutions. So I think this will be a hint for you to think that, oh, using one glass rod, it can cause contamination, right? And the liquid in each beaker was then stirred for 10 seconds. So stirring for 10 seconds, it could consider as a constant variable, as well as imagine, right, you as a student or you as an experimenter doing 4 different pieces, or let's say 2, 3, bio, non-bio, and water, right? And you were stirring, and that might not be consistent, right? So it can also lead to the inconsistency in the stirring, and the liquid in each beaker was stirred again for 10 seconds after 5, 10, and 15 minutes. And after 15 minutes, so it means the time is constant, and each piece of cloth was removed from the liquid and placed on the label area of the white tie. And step number 7, again, the student observes. So whenever a student or an experimenter observed on the result, it could lead to the subjective error. I don't want to say error, but it's more like a subjective because I may have different eyesight to the other people, right? What I mean is that I may see one color as blue, but other student or other people may see that, oh, it's light blue, or some people might say it's sky blue. So it's me, different people have different eyesight, meaning that it's a subjective problem, subjective error, and they use the intensity score in the table 1.1 to determine the intensity of the stain on the 4 pieces of cloth. And the result were like showed here. So the 4 pieces of cloth at the end of the experiment it's shown here, and then they give you the intensity score as well. And for the first question, they ask you to prepare a table to record the result in the space provided. So this is a table. So we have two column and like 5 rows. So we have piece of cloth and intensity score, bio 1+, non-bio 2+, and water not washed. And then like you go according to the intensity score provided, and then by just looking at the intensity of the result provided there. So this will be the first question. So I would like to suggest you to have a plan before making a actual table that you want to use, so that you will not messed up, or it will not be a dirty piece of paper, right? So make a plan first, and then like you draw using a pencil and a ruler for the outline of the table. So it will make it easier for you to erase if you make some mistake or some error, and then only you start to write it. And in this case of questions, there is not much about unit, but if they contain, or if the question is a question involve unit, make sure to include it in the title. So for example, let's say temperature, and then put in bracket degree Celsius, or distance in bracket centimeter or millimeter. What I mean is that don't forget to put the unit in the headings, right? So that would be the table. And then they ask you about the dependent variable. So throughout the questions reading, we already state that dependent variable is the intensity of the stain. And one variable, so they only ask one variable, but we identify at least two variable that were kept the same, right? So I chose to answer the same type of food, or like the same type of stain they used would be the one variable that was kept the same in this investigation. And for question number 4, it's asked to identify one source of error in step 4, right? You see that they ask you about error, and then you need to explain how it might affect the result. So here, explain. So error would be using only one glass rod, or we can say like one unwashed glass rod, right, for each solution, and it can lead to contamination. So here I wrote down it, but my T is a little bit of different. Okay, so here it can lead to contamination. And then identify a control in this experiment. So it have to be in this experiment already, and explain why this control was used. So in this case, you can see we have four different pieces of cloth used: bio, non-bio, distilled water, and not washed. So you can see that only bio and non-bio are included as the independent variable, right? But we got two, which are distilled water and not washed. So both can be identified as a control. So here I chose to answer a piece of cloth in distilled water, because control is to compare, right? So this is used to compare the effect of the other treatment. So to compare, like, oh, see here, this is without stain, and you can say any other blah 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 result, right? So it will be the distilled water and also not washed. And number 6 is step 7 may not give accurate results. So you need to state why the method used in step 7. So as I mentioned already, in step 7, the result were observed by student, which is subjective, right? Since different people may have different eyesight. So by seeing, or like by observing with our own naked eye, may not be very accurate. We don't want to say that it is not a very bad result, but it may not be very accurate. So this would be the methods that were used in this experiment, which can give inaccurate result. And then continuing to question number B, some student wanted to find the optimum, or like best temperature for using biological washing powder. So here we said like by using different temperature. So that would be the independent variable, and they use agar jelly containing starch, circular holes, and also solution of biological washing powder for 10 degrees Celsius for 10 hours, and they also use the iodine solution. So you can see that they use a agar jelly which contains starch, and also they have iodine solution. So to test the presence of starch, right? So the result already given here in the figure 1.2. So agar stained pale brown, it's mean negative starch. And agar stained blue black, it's mean positive result for the starch test. And the question asks you to state the independent variable. So in this case, it is temperature. And then after looking at the result provided, you have to state a conclusion for this result. So you can see that a blue blood stain indicates that the starch is still present. Or on the other hand, you can also mention that the pale blue stain indicate that the starch is absent, right? So it's mean that the washing powder solution diffuses, right? Diffuses. So you can mention like that. So that would be the conclusion. Okay, and then number three, I would say is the most easiest one to gain the answer if you pay attention to the questions and if you do it correctly, right? So here they give you a diameter of the pale brown stain area was measured during the investigation and result provided. So they ask you to draw a line graph. So in biology, usually there are two types of graph only, so which can be a line graph or a bar graph. So make sure you read or you pay attention to the questions carefully so that you gain full points, right? So when we draw a graph, there is an acronym that we can use to apply to check whether your graph can gain full score or not. So in short form, it is SLAP, right? SLAP, S-L-A-P, SLAP. So S indicate for scale. So you need to use the scale correctly and make sure you use the graph more than half of the space provided, meaning that your graph should be large enough, not a very tiny small one at the end of the corner, right? No, you have to use the scale more than, you have to use the graph space provided or the grid provided more than half of the grid provided. And number slapped L would be the line. So make sure you look at it is like line graph or best fit line, and A stand for axis. So you have to correctly state the x-axis and y-axis. So I would suggest that usually, so x-axis would be independent variable, which in this case we have temperature. Oh, what I mean by unit in the previous questions, right, from the table drawing, what I mean by the unit to include is here, temperature, you have to mention degree Celsius. And then here, distance. You are not putting every single temperature like this, right? We don't use like this. This is no, it's not recommended. So you have to write every unit in the heading. So we got axis, and P is point correctly plotted. So you have to point the graph, you have to plot everything correctly, and then you draw a line using a ruler and a pencil. So I would suggest you using a pencil and a ruler so that it will be easier again to erase if you made some mistake or like some error while drawing a graph. So here we got temperature on the x-axis and diameter of pale brown stain by millimeter on the y-axis. So it's not that really difficult because you can just copy everything from the table provided, right? So this would be a graph drawing for this paper. And then describe the trend. So they ask you to describe the trend shown by the data in table 1.2, also from the table 1.2 as well as from your graph. So by observing the graph and the table, you can see that as the temperature increases, the diameter of pale brown stain area also increases. And then for a certain period, let's say certain degree, like 10 degrees Celsius, it still remain constant and then it decreases, right? So that would be the trend. And then you don't forget to use the data provided. So here, this is just my sample answer, right? So as the temperature increases from 10 degrees Celsius to 40 degrees Celsius, the diameter increases from 11 to 22 millimeter. Then it's remained constant at 22 millimeter till 50 degrees Celsius. And after that, it decreases to 5 millimeter at 70 degrees Celsius. Okay, that would be just my sample answer provided. And number 5, the student were unable to determine the optimum temperature. Yes, obviously, because from the graph, even though it may be seen this range might be the optimum temperature range, but we cannot be sure whether it is 40, 41, 43, 45, 46, something like that. We are not very sure. We cannot be able to indicate a one temperature that can be considered as optimum, right? In this case, it is in the range of 40 degree and 50 degrees Celsius. So describe what additional measurement would be needed to find the optimum temperature. So the student should measure the diameter at smaller interval between 40 degrees Celsius and 50 degrees Celsius, meaning that instead of 10 degree interval, students should use, let's say, one interval or two interval. One would be better, but so 40, 41, 42, 43, 44, 45, 46, that kind of smaller interval would be better to give the more accurate result. So this would be the end of question number 1, which was 20 points. And question number two, they show you about a photomicrograph of lung disease at the same magnification. One show of healthy lung tissues and other one with the COPD. And you can see from the figure, so one have healthy and another one is with COPD. And number 1A asks you to make a large drawing of the lungs diseased of the person with COPD shown in figure 2.1. Do not draw individual cells. So in this kind of question, they are testing you to draw the outline. You don't need to shade. You don't need to draw an individual cell. So outline is important. And the size, make sure you draw something larger than the provided diagram. Some students, they make mistake that their diagram is even smaller than the provided image. So that is not a large drawing, right? So make sure you draw something that is larger than the provided diagram and pay attention to the details. So in what I mean by detail is that make sure you trying to manipulate as similar as possible, right? As identical as possible. Don't just draw something you think, oh, this is the same, right? So make sure outline, size, and detail will be important. And for number two, I would say sorry for those watching my video because I'm doing on my tablet and I cannot measure the correct measurement. So I just skip it the whole thing. But in this case, please you go measure the length of CD and also the length of CD from your diagram and make sure you use the same unit. So what I mean by this is if you used centimeter, or the best one would be millimeter. So if you use millimeter to measure the length of CD on the diagram provided, then use millimeter for the length of line on your drawing as well. So make sure the unit are the same. And also after calculating, they ask you to give you the nearest whole number. So don't just leave it with one decimal, two decimal. Make sure you use you use the answer which has the nearest whole number. And number three, describe three visible ways that the lungs of the healthy person differ from the person with COPD. So from the diagram, you can see that the healthy person, the lungs of the healthy persons contain more alveoli and also they have a thinner alveolar wall, so which provide large surfaced area as well. And also you can see the additional information in the healthy person, well, they contain the bronchioles and as well, right? So, but in this case, I mentioned three visible ways. So make sure it have to be visible. You cannot write something that you learned from the class, right? So it should be visible from the diagram. So the healthy person lungs have more alveoli and it has the thinner alveolar wall and also it have larger surface area. So it should be in terms of comparative form because you are comparing between a healthy person and a person with COPD. And then number B, to investigate the concentration of carbon dioxide in expired air compared to in inspired air, right? So inspire and expire air would be the dependent variable. Oh, sorry. So expire and inspire air would be the independent variable and concentration of carbon dioxide would be dependent variable. So in this case, they ask you to state a one possible hazard. So in this case, when a student or when the participant inhale or exhale very strongly, the lime water or the indicator solution might get inhaled. Okay, and number two, one substance beside lime water. So it will be hydrogen carbonate indicator solution. So remember, there are two indicator solution to use to detect or determine the carbon dioxide concentration. And then this question asks you to calculate the concentration of carbon dioxide in expired air. So they give you in test tube A, it took 600 seconds for the lime water to go cloudy. So which means it take a longer duration, right? Because in test tube A, it's inspired air. Inspired air contain less carbon dioxide. But for the test tube B, it's contain expired air and which contain more carbon dioxide. And it's took only 6 seconds, so which is a very fast rate to turn the lime water cloudy. So to able to calculate, so I start off with finding the ratio of carbon dioxide concentration in test tube A and test tube B. So which I would get 100, and then I multiply with 0.04%. So I got 4% in the expire air. So which I think align with our learned lesson from the lecture, right? Where we got 0.04% in inspire air and 4% in expire air. And for the last question, which you have to write for 6 mark question. So a student wanted to investigate the hypothesis. So a student stated that expire air contain more carbon dioxide immediately after exercise than before exercise. So from this, you have to plan an investigation. Again, remember, in this kind of questions, you are not expected to give the results. So you just need to give a plan throughout investigation plan for the examiner, like how you would carry out this kind of experiment. So in this case, independent variable would be before and after exercise, and dependent would be concentration of carbon dioxide. But we cannot just take concentration of carbon dioxide. It should be time taken for lime water to turn cloudy. And for the constant variable, you should use same volume of lime water, same intensity or same duration of exercise, same duration for like resting period. And in every experiment, make sure we have to include a repeat, which would be at least, let's say, 3 to 5 participants to make sure that we got a similar result or we get the accurate result. And for the CIE practical, the planning investigation plan questions, you have to include a safety precaution. So in this case, we are using the mouthpiece, so where the participant breathe, inhale, and exhale. So we have to sterilize the mouthpiece every time we used, and also to refresh the equipment before the next measurement. So this is not the answer, but you have to give in paragraph form, and like you have to write this in the paragraph form. This is just the outline I have provided as a sample. So this would be the end of this paper and also end of this video. So thank you so much for watching the video and hope to see you all in next video. So thank you so much. Have a great day all. Bye-bye.